Guys, this is your learning assistant for Biology 1009. Today I have a short video for mutations. So mutation is referring to a heritable change in the genetic material. There can be one positive and there are like a positive and negative sides for mutations. So for the positive side, mutations are the foundation for evolutionary change that is needed for a species to adapt to changes in the environment. But on the other hand, the negative side, new mutations are much more likely to be harmful than beneficial to the individual. So mutations often cause the disease disorders, a lot of different bad consequences. So more likely, mutation will cause negative results. There are three main types of mutations, and the first one is chromosomal mutation, which is the changes in chromosome structure. The second one will be genome mutations, which is the changes in chromosome number, like chromosome 9, 22. Those numbers will be changed. And the one that I'm going to talk about is the gene mutation. It's relatively small change in DNA structure that influences a single gene. So there is a gene mutations usually involve a point mutation, which is a change in a single base pair, and point mutation has like 4 different types, and those are the main things that I am going to talk about in the video today. So the first one is a silent mutation. It's a base substitution that does not substitutions that do not alter the amino acid sequence of the polypeptide. So it's due to the degeneracy of the genetic code, meaning one amino acid can be coded by multiple, like 3 or 4, whatever, multiple codons. So for an example, like a proline, and a type of one of the 20 amino acids, proline can be coded by like CCU. Let's say it can be coded by CCU, CCA, CGC, like that. So like even though there's a different base substitutions by silent mutation, because they all of them code for one amino acid, nothing changes. Like there will be no signs of alteration. But for missense mutation, it's a whole different thing. So it's a base substitution in which an amino acids do change. So for an example, it's like a sickle cell anemia. Right here, I have a picture. So the normal red blood cells have the donut shape. But then when missense mutation happens, a base is switched up into a different thing, which causes the normal red blood cell to become a half donut shape, which makes the red blood cell to lose the function of transporting the oxygen. So there will be a malfunction in blood vessel for transporting the oxygens. Therefore, a human body will have a lack of oxygen, causing causing the sickle cell anemia. So sickle cell anemia is a great example of missense mutation. Nonsense mutation is pretty much a mutation that causes a premature stop codon because it changes this normal amino acid into a stop codon. So like stop codon due to nonsense mutation can happen right after start codon, or it can happen after like a few number of different codons. So it doesn't matter where the stop codon happens. If there's a premature stop codon, then it's pretty much a nonsense mutations. And the last one here is the frameshift mutation. It's involving the addition or deletion of a number of nucleotides that is not divisible by 3. So this shifts. So the frameshift mutations shift the reading frame. So the translation of the mRNA results in a completely different amino acid sequence downstream of the mutation. So yeah, this is it. So this is what I have for the mutations. Thank you.